This is the research update for 2016 from the Chromosome 18 Clinical Research Center. We want to share the progress toward our goal of making the chromosome 18 conditions the first treatable chromosome abnormalities. Just to briefly review those conditions, there are 5 general groups of chromosome 18 conditions, within which there can be a huge amount of chromosome variability. As you've likely heard me say many times, anything you can do to this picture of chromosomes with scissors, glue, and a copy machine can and does really happen. It's just that most are not compatible with life. The chromosome 18 conditions are the ones that just happen to be more survivable to birth. Shown at the top of this slide is trisomy 18. This is by far the most common of the chromosome 18 conditions. But because the needs of these families are so different, and because there are other organizations focused on trisomy 18, we have devoted fewer resources to this condition. Although we are still interested in trisomy 18, it's just not our area of emphasis. Our focus has been on the other conditions shown here. On the upper left is the 18q- group, which includes anyone with a deletion of one or more genes from anywhere along the long arm of chromosome 18. These deletions can include up to 100 genes and are most commonly from some breakpoint to the end of the chromosome. But alternatively, the deletion can be from the middle of the long arm. Every person with an 18q deletion has a unique deletion. Going counterclockwise on the lower left is tetrasomy 18p. People with tetrasomy 18p have an extra chromosome, but that chromosome is made up of 2 short arms. The result is a total of 4 copies of the short arm of chromosome 18. Almost everyone with tetrasomy 18p has the same chromosome content of their extra chromosome. So in this case, this is a more genetically homogeneous group than 18q-. Next, on the lower right, is 18p-, in which people have a deletion of some portion of the short arm of chromosome 18. About half of the people with 18p- have a deletion of the entire short arm, making them a homogeneous group. But the other half of the people with 18p deletions each have a unique breakpoint, and therefore a unique set of genes present in 1 copy instead of 2. On the upper right is ring 18. As you can see in the diagram, one of the chromosomes forms a ring, which by definition means that there is some missing chromosome material from the ends of both chromosome arms. But the amount of missing material varies from person to person, from a deletion that includes no genes to one that includes nearly 100 genes. This makes this group of people very heterogeneous because of the variability in size of the deletion from each arm of the chromosome. We are often asked how common the chromosome 18 conditions are. When you consider all chromosome abnormalities, they in total are actually quite common, and the numbers are shown here. There are different ways you can think about the same prevalence numbers, so we're explaining them in 2 different ways here. There are 438 babies with a chromosome abnormality for every 100,000 babies born, or 1 baby is born with a chromosome abnormality for every 230 babies born. These are, however, not perfect estimates. The problem with these numbers is that they come from looking at the medical records of a large number of children in national health systems, and they're asking how many of those children are diagnosed with a chromosome abnormality. So this number does not include children who have a chromosome abnormality but were not diagnosed. They may not be diagnosed because they had milder issues, or more likely because they received substandard care. This means that this may not be a very accurate number, but it's the best we have. Within this group of chromosome abnormalities, just over half have trisomy 21 or Down syndrome. The other half have any of all the other possibilities, 5p-, 11q-, ring 18, etc. And because we had a speaker at the conference talk about the fantastic work of the Cystic Fibrosis Foundation, I've added cystic fibrosis as a comparator rare disease, but it is not a chromosome abnormality. Keeping these numbers in mind, now let's look at the individual chromosome 18 conditions. The numbers from the last slide are shown at the bottom. Trisomy 18 is the most common, with 58 babies out of 100,000 having trisomy 18. Or stated differently, 1 out of every 1,710 babies born has trisomy 18. You can see that it is far more common than the other chromosome 18 conditions. 18q- occurs in 1 out of every 54,764 births, 18p- in 1 out of every 56,336 births, ring 18 in 1 out of every 300,129 births, and tetrasomy 18p is the most rare with 1 out of every 625,000 births. But again, these numbers are derived from medical records, and some individuals may be undiagnosed. The right way to get these numbers would be to do a chromosome analysis on a very large number of consecutive births, say 100,000 or 1,000,000, but that's cost prohibitive at this time. I often have people ask me why there's not a chromosome 18 clinic in their local medical center. From this, you can see that the numbers just can't justify it. For every child with a chromosome 18 abnormality, there are 100 with Down syndrome and 10 with cystic fibrosis. This is all the more reason why we need to collect all the available information in a single place so that we can have any meaningful insights. Then we disseminate those insights to the families' treating physicians. This is just a reminder of where we started. Here are the pages for 18q- and 18p- from the primary source book of the day, back in the days when books were the go-to source of information. The information on these conditions was primarily focused on how to recognize and diagnose the conditions. So things like small stature, obnoxious behavior, and wide mouth were key points. There was very little functional information and no therapeutic guidance. In contrast, our goal is to make the chromosome 18 conditions the first treatable chromosome abnormalities. By this, we mean the goal is self-sufficiency. It's not to turn them into different people, but it's to enable them to grow up to be the masters of their own lives. For parents, this means they won't need to think about special needs trusts, estate planning, and who will care for their adult child when they're gone, just like they don't worry about those things for their other children. For the affected individuals, it means they can decide where they live, who they live with, what their employment will be, and not have to settle for the choices made by someone else who thinks they have their best interest at heart. Can we do this? Of course we can. It's just biology. It's not fate. It's not bad karma. It's not the punishment of the gods. It's just biology. And if it's just biology, we can figure it out. And this brings us back to what it's all about. These amazing people with chromosome 18 conditions. Because every child deserves a chance to succeed. Because these are such rare conditions, and because so many different medical subspecialties need to be involved, we've chosen to centralize the information and expertise in a single location. The Chromosome 18 Clinical Research Center is located in the Department of Pediatrics at the University of Texas Health Science Center at San Antonio. The first thing any family has to do in order to be a part of the research study is to complete the primary enrollment. You can't participate in any of the surveys or clinical evaluations until you've completed the primary enrollment. This includes signing consent forms and sending copies of medical records. Once we have those things, we will send a blood kit for the collection of blood from the child and both biological parents. At this point, every family will get a unique study number and then be eligible, but not required, to participate in other projects, such as surveys or clinical evaluations. The exact nature of these other studies changes and are dictated by the nature of the important research questions that we're trying to answer for you. In a new study that's being set up, say, for a clinical trial of a new therapy, only those families who have completed the primary enrollment will be offered the opportunity to participate in that study. We also collaborate with other researchers at other institutions when they have expertise that can help us. This expertise may just be data analysis, or it might be the expertise on the evaluation of a function of a certain gene. In these cases, the investigator only receives your study number and does not have any personal identifying information about you or your child. They would never be contacting you. We have collaborators around the world helping us to understand certain aspects of the chromosome 18 conditions. These are our current enrollment numbers. Considering the rareness of these conditions, this is an astonishingly large number of families committed to making a difference. And as you can see, these families come from all parts of the world. The core research center activities include the DNA and cell bank from all the blood and skin samples we've collected, the medical records and clinical data database. The survey-based information provides us with longitudinal and developmental data. We also do the DNA analysis on all the participant samples, as well as clinical evaluations when such studies are being conducted. And of course, we provide information for families, from the results of their own clinical evaluation, to general syndrome information, to responding to individual questions from families and their physicians. Because the medical issues are diverse, we have a large group of collaborators from a broad array of medical subspecialties. In addition to these clinical investigators, we also have collaborators who work on the mouse models, who are expert in mouse behavior analysis and mouse electrophysiology. In support of all the investigators, we have a group of professional staff who keep the whole thing moving forward. We have Gloria Matthews, who's our administrator, takes care of all the budgeting. Patty Hurd runs the lab. Research staff include Annis Hill, who's the project manager, Mimi Hasse, who's the patient navigator and abstracts all the medical records. David Rupert is our geek, takes care of the databases and computer issues. Bridget Swallow is a psychometrist, and Courtney Siebold is our genetic counselor. And those of you enrolled in the study have probably corresponded with any or all of these individuals. This is the same slide I've used for many years to explain our approach. The first two are parallel activities, both leading to the third. But let me explain. At this point, there's not time to go into everything we've learned and everything we're working on with regard to each of these for each syndrome. But instead, I'd like to highlight just a few things for each syndrome in each one of these categories. First, defining standards of care. By this we mean taking these individuals' medical concerns seriously. Not assuming they're too messed up to help, and not assuming that since you can't correct the chromosome abnormality that they can't be helped by standard medical approaches. With regard to 18p-, we investigated their slow growth and determined that many were growth hormone deficient, which is a treatable condition. We investigated their delayed speech and found that many were hearing impaired. In future work, we're particularly interested in learning more about the autism spectrum in this group, as well as understanding the nature and progression of the speech dysarthria and autoimmune disorders that seem to be developing in some adults. These are not comprehensive lists, but just a few highlights. With regard to 18q-, again, we actually evaluated their slow growth and found them to be growth hormone deficient. And as I said before, this is a treatable condition. We assess their cognitive parameters to learn where they do and do not have the most trouble. We evaluated and provided guidance for such things as hearing and myelination. In the future, just to name a few, we're particularly interested in learning more about autism, depression, and the development of autoimmune disorders as they get older. And of course, all the 18p- and all the 18q- information informs those with ring 18. With regard to tetrasomy 18p, both past and future work involves the identification of bone density issues, followed by understanding the cause and finding a treatment. We have also published on the cognitive and behavioral profiles of people with tetrasomy 18p. And in the future, we're very interested in learning more about the autism spectrum aspects of their condition. The information on the standards of care is disseminated in multiple ways. There are, of course, the syndrome descriptions on the Chromosome 18 Registry website. But we also provide information to other sources, including Wikipedia, the Genetic and Rare Disease Information Center at the National Institutes of Health, and to other organizations such as Unique. In addition, we're about to publish management guides for physicians who care for individuals with the chromosome 18 conditions. And as always, you can ask the experts. If there are questions, just email Annis Hill, and she'll see that the right person answers your questions. Her email is shown here. The second component to our approach is to identify key genes. This has been ongoing since the beginning. First, by defining the exact gene content of each person's deletion or duplication, and then by identifying regions of the chromosome that are deleted or duplicated in people with a particular medical aspect. For example, identifying a region of the chromosome deleted in everyone with 18q- and closed ear canals. This helps narrow down which gene might be responsible by narrowing down the region associated with that condition. Today, the function of many genes are being determined by diverse research groups around the world. When information becomes available about the function of a gene, as well as information indicating that it could be one of the few genes that can cause a problem when there are too few or too many copies, then we investigate further. At least once a year, I look up all the information on every one of the 264 genes on chromosome 18 to see if there might be some information that would help us. Over 70% of these genes have information that leads us to believe there are not adverse consequences from having an abnormal number of copies of that gene. Some genes have no information on which to base a conclusion. But this leaves a very small number left to investigate. This is very much the opposite approach from defining standards of care. In defining standards of care, we began by evaluating the participant to understand how best to treat. In identifying key genes, we use the knowledge of the gene function to investigate its potential impact on the person. Those of you with 18p- may recall that a couple of years ago, we had new information on several genes on 18p that led us to suspect that they could cause health issues in someone with 18p-. We investigated the possible impact of these genes by sending surveys and requesting medical evaluations. These studies are still ongoing as we try to determine if these genes do in fact have an impact on someone with 18p-. Future work involves trying to understand the potential implications of too few or too many copies of the mLN2 gene and its impact on blood clotting. For 18q-, just to name a few, we identified MBP as important in sensorineural hearing loss and the SETBP1 gene as important in expressive language. Ongoing and future work involves trying to appreciate the physiological impact of a TCF4 deletion on brain development and an MC4R gene deletion on weight. In addition, we're also investigating the role of specific genes on behavior and cognition in mice with deletion of these genes. This information can provide the initial insights into what to look for in people with a deletion of that gene. Tetrasomy 18p has been a bit more challenging in this regard because everyone has the same duplicated genes. We thought gene expression studies might help identify the genes dysregulated by a duplication. But we found too much variation between different individuals to be able to detect meaningful differences due to the duplication. So our next approach is to identify people who are mosaic for tetrasomy 18p and get skin samples from them. Then we separate the normal cells from their abnormal cells and then look at the differences in gene expression. But this is a project in need of funding. In addition, we're also looking at the effects of the mLN2 gene on blood clotting in people with tetrasomy 18p. This project does have funding. The final and ultimate is changing gene action. We're just beginning to be at a place where we can explore such activities. When there's not a standard clinical treatment for an aspect of the chromosome 18 condition, and when we know which gene is the cause, then we can begin to try to change the activity of that specific gene. This is where the mouse models come in as a first test for the effectiveness of new and untried therapies. We have a couple of these projects ongoing involving the NETO1 mouse. NETO1 is a gene on 18q that we think has behavioral effects. To summarize where we are in this quest to make the chromosome 18 conditions the first treatable chromosome abnormalities, we have 12 projects in the data analysis phase, examples of which I just talked about. In addition, there are 8 projects in the data collection phase. Some are surveys and some require getting additional samples, such as skin samples. Then there are projects that are stalled for lack of funding. These include clinical evaluations. In particular, we need to do clinical evaluations of adults with 18p- because as they're getting older, they're developing new health problems. We need to understand those issues so that we can determine the best course of treatment. In addition, there are additional mouse models for other key genes that need to be investigated, and there are additional DNA analyses that need to be performed. This is a list of our publications for the last year. You can find all of them on the website for the Chromosome 18 Clinical Research Center. And most of these have a short summary for parents about the importance of that paper. Even though we do not have funding to bring in individuals for research evaluations, we do recognize that we have a lot of expertise here in San Antonio. So if families want to come at their own expense, we can help that get arranged. For people not enrolled in the study, they can get the names of the physicians from our website. For those who are enrolled in the study, Annis can help arrange your visit so that you can get the appointments within a few-day period of time. She can also advise on hotel and other local arrangements. The reason we only do this for study participants is twofold. First, it's a way to give back to the families who go to the trouble of filling out surveys and staying in touch with us, and we appreciate that. Also, because we have their medical records, we can advise families on who they should see, and we can give copies of the relevant past medical records to each physician before their appointment. The process is shown here. First, contact Annis to discuss possible dates, specific questions, or concerns. She will be sure that we have updated medical records as well as health insurance information. She will make recommendations on which clinicians you should see. Annis will work with the clinic and the family to make appointments. Then, at each appointment, we will ask you to fill out a release of information form so the information can go back to the research center and not just go into your medical record. This is different from a research study visit in the following ways. You and your insurance company pay for the medical costs. You pay for your travel and housing. And the research center does not automatically get the information. You have to fill out a release of information form for each appointment. And the reports from these appointments go to your doctor, not directly to you, just as they would from any other doctor visit in your hometown. The research center budget is now cut to the bare bones. This is why so many projects are on hold. Other sources of funding are just no longer available, and the registry support is all that sustains the center. Most staff members are also working part-time for other investigators. The 2013 budget of just over $500,000 is what was necessary to be operating at a baseline level so that we have the ability to sustain enrollment and DNA analysis as well as get preliminary data for new ideas and projects until those projects can get separate funding. The current budget for this fiscal year is $300,000. The current research funding environment is bleak. The success rate for research grants to the National Institutes of Health, National Institute for Child Health and Human Development is 9%. This means that all things being equal, you have to write 10 grants to get one. Since it takes at least a month to write a grant, that means 9 months of time totally wasted. So this is not a good use of my time, especially since a single grant won't solve all our financial issues. It would just fund one of the many projects on the table. Recently, we attended a 2-day symposium on Down syndrome. The most exciting work was presented first, and by noon on the first day, none of the presenters had that work funded by the National Institutes of Health. It was all funded by philanthropy. By the end of the conference, only a very small minority of the work presented was funded by the NIH. Chromosome abnormality research, even for Down syndrome, is just not viewed as important. At some level, this makes us relieved that the problem's not just us. But on another level, it's very discouraging that conditions that so profoundly impact so many people are not considered important. And when you consider that for every person with 18p-, there are 125 with Down syndrome, we have to realize that it's up to us. The only way any of this will happen is if we fund it ourselves. Just as a group of examples, here are the costs of some of the things that we do or purchase for the research projects described here. Doing it right costs a lot of money. Thank you for your attention, and thank you for caring about the chromosome 18 conditions. As always, if you have any questions, please send those through Annis at this email address. She will harass us until we answer you. Thank you.